Okay, so the webinar today, we're going to be discussing fair data and you, and really, I think the byline to this: are you ready for 2023? So there are new mandates coming out that start in January, and so basically, we'll be going through what's needed for probably your next NIH grant submission, unless it's in the next 2 months. But if you're submitting in January, there are some new things that you will have to know. So first, a little motivation. So a lot of discussion around data sharing, fair data, links back to open science and the open science movement. And really what that is about is really, dissemination of all types of information around science. So the papers, so the open access papers that you get through PubMed Central, and what the White House has recently strengthened is the ability to get access, open access to the scientific literature as soon as possible. But it's also about the open access to data as well, and so that's something that has been strengthening over the last number of years. And especially during COVID, there was a lot of work done remotely. There's a lot of data sharing around COVID, and for example, the genomic sequences that were made available. And so there's a lot of discussion around how COVID sort of pushed sort of open science and open access to the literature and data. And there are a number of motivations for open science. I mean, it's just a consequence of the digital revolution, right? So the access we have to larger and larger amounts of data, and the access to computational services we have to be able to go through large amounts of data. There are other arguments for biomedicine. One of them is that research is largely funded by the public, and the public should have access to the results. And various aspects of open science which lead to better science. So the idea of transparency, so the ability of multiple people to look at the papers, the data, more eyes on the data, and the increased computational capacity can lead to the mining of the literature and data for new insights. And there are sort of two aspects of data as well. So we all know about sort of the big science projects that generate large pools of freely available and usable data, right? So there are a number of large data collection activities that NIH funds. But there's also what we call sort of the long tail of data, right? So this is all the data, underlying claims in papers in the literature, and that is something where this is going to become now sort of this published data will become a primary product of research itself. And that's where we get to sort of NIH's new mandate that takes effect January 25th, 2023. Basically the high level takeaways are that all data must be managed and most data should be shared, and that this data should be as open as possible, as closed as necessary. And this is going to be monitored in the sense that you now have to include a data management and sharing plan that is going to be approved by the program or scientific officer for funded proposals. But really, they want you to now to more fully describe the data management portions as well as the data sharing aspects. Now, again, the effective date is for any proposal submitted January 25th or later, or for any contracts that are sort of executed on January 25th or later. So this is coming in a couple of months, and it defines what it considers to be scientific data, right? So scientific data Is the factual material, what you either record, from your recording devices, or what you record in your notebook, um, you know, that is used to generate the research findings. And the important aspect here is, regardless of whether the data are used to support scholarly publications, right? So you may have collected, let's say you're dealing with a number of subject populations. You've collected data for 3 of them. One population didn't quite work out. There was no significant finding, so you published on 2 of the other subject groups. But this mandate requires the sharing of that third group of data. And that is sort of what we normally call dark data, right? This is data collected in scientific labs, in research labs, that never really see the light of day in a publication. And that's because, maybe the hypothesis didn't pan out. And so, publishing, negative results is not common practice. But in the case of this, of the data itself, this policy does require the sharing of that data. Now, in terms of the definition of data, scientific data does not include, the laboratory notebooks, the any preliminary analysis, drafts of scientific papers or plans for future research. So data has a specific meaning in the context of this mandate. And again, important point, even though scientific data not used to support a publication are considered scientific data and within the policy scope. So that is a very critical thing to remember. And this is new, okay? In terms of prior data sharing mandates for NIH and also for journals, it was in support of publications, that have been accepted and published, that you'd have to include, the supporting data for that. The when is also new in terms of what's required. So basically, the scientific data should be made accessible as soon as possible and no later than the time of an associated publication. So again, if you have data that supports a publication, that must be shared at the time of publication, or data that may not be associated with a publication, right, must be shared at the end of the award support period, right? And this is whichever comes first. So if you have data that you're also working on a publication for, and you're coming to the end of the support period, you have to be careful to make sure that you meet the mandates. And then, the where, NIH encourages the use of established repositories to the extent possible. How you're supposed to share data, is that the data management and data sharing practices should be consistent with the FAIR data principles. And I'll talk a bit about FAIR in a few minutes. And then an additional sort of new aspect is that the funding. So the fees for the long-term data preservation and sharing are allowable. And the key thing here is that usually in NIH grants, you're not allowed to pay for things that go beyond the limit of the current grant year. But for the data sharing and data preservation fees, one is able to spend those fees during the performance period, even if the scientific data and metadata preservation that's being paid for is beyond the award period. So this is something new as well. At the end of the funding period, non-compliance with the plan could be taken into account by NIH for future funding decisions. So they are putting some teeth into this in the sense that the program officers that are managing these awards will also be involved in looking at compliance with data management and sharing that is outlined in the proposals. And this policy applies to all research funded or conducted in whole or in part, right? So that, if you have a project that's only partially funded by NIH, these rules apply to all data that are collected as part of that funded research. So again, this is the new policy that takes effect. The one sort of important takeaway, from the renaming of their data sharing plan. We used to have a resource sharing plan, a data sharing plan. They've now added that it's data management and sharing plan, because the best way to prepare yourself for the ability to share the data is if you have good data management practices, from the get-go. So there are a number of ways that you can do this. Here's an article from 2018 that talks about data management for researchers. So again, having standardized procedures for the management of data before you even need to share the data is critical to be able to ensure that the actual sharing process isn't too onerous. So this is all part of the changing culture of around data management and sharing. So this is, again, the data management and sharing is not just for others, right, and science and society. It's for you yourself, right, for managing your data in the lab, being able to work efficiently with your data, working with students in your lab who might be using the data. It's for the future self, 2 years, 3 years down the road, you're doing a new study, but there's something that you need to look at potentially from some data that had been collected prior. How can you actually find that data? How can you actually reuse that data yourself? And then also, if you're collaborating amongst a couple labs in a project, right? So good data management practices help in that as well. So it's not just, again, good data management is not just for data sharing, but for all your sort of research activities. So how do you do that, and what are the FAIR data principles? So a little background on FAIR. So the FAIR guiding principles on data management and stewardship have 4 high-level principles that data must be findable, accessible, interoperable, and reusable. And this is for humans and machines. So again, as we go to the use of more AI, machine learning techniques, we need to be able to allow the computational processes to also have access to this data. And so, machine readable, again, means that the metadata and data is in a format that can be easily processed by a computer without human intervention, right? And so there are a number of standard formats for this, where, that type of data can be marked up. And, the other aspect, of data, one of these is actual table that one can extract information from. The other is a JPEG image, right? And so when sharing data, sometimes in supplementary material where people have shared data, they include images of their tables, which is not sufficient in terms of making sure that that data is actually readable. And so again, that is important when sharing data. But now looking at sort of the 4 principles of FAIR, if we look at findable, basically, the 2 important aspects here are that that the data are assigned a globally and unique persistent identifier, and that the data are described with rich metadata, right? And so what does persistent identifier mean? We're all familiar with a number of these. So for example, when you publish a paper, you get that DOI, the digital object identifier. That is the persistent unique identifier for that paper. You all have ORCID. So, a number of journals, NIH is requiring the use of ORCID. That is your unique persistent identifier for yourself, in the research sphere. There are also resource identifiers, which, identify antibodies, model organisms, and the like, right? And so again, these are unique identifiers, and they can't be reused. So unlike, website URLs or catalog numbers that may change, your digital object identifier for your paper will never change, right? Your ORCID will never change. And these are issued by official registries who track and identify, these unique entities. And, the important lesson here is you can't assign PIDs to your data yourself, right? And so these identifiers you get through the depositing your data in a trusted repository. And these trusted repositories provide those identifiers, just as the journals provide the DOIs for the journal articles themselves. So, what are rich metadata? And this is data about the data, which includes file format, file size. So there's sort of the sort of core data, that describes, the various data artifacts, but there's also a whole host of descriptive metadata that describes the resource, for use in discovery. But it also, includes, this is the standard stuff you have with journal articles, like the title, abstract, author, keywords. But then you get into more study-specific items like the organism, the condition studied, additional metadata, that is needed, to fully describe, the data set. And, again, with papers, we do know standard structure, standard metadata. We're all familiar with this. We all know where to find the introduction. We all know where to find the method section. And so, these things together, provide a way to richly structure the publication itself. And the same thing can be done with data sets, right? So you have the same information, for example, that is related to, the publication, but then you also might have additional things like links to experimental protocols, for that specific data. You have usage notes, informations on subjects and samples, links to potential code that were used, in the analysis of the data. So again, the richer metadata, to describe that data. And this is driven also by repositories where you may be depositing the data itself. So again, one thing about metadata, more metadata is better than less metadata. I've never run into someone that said, oh, I wish I had, not assigned as much metadata as I was collecting this. So again, you can think of metadata as notes in lab notebooks as well. You're describing, what is going on in the experiment. But I have run into situations where people have not written down enough information, and when that information is forgotten, it is lost. And so that is critical. So the A in FAIR is accessible. And so again, this is that data are retrieved by their identifier using a standard communication protocol. And so this is, again, driven by these repositories. So if you share your data through a community repository, then this is taken care of for you. And they also provide, structured metadata. So this is repositories will have a data model that sits behind the repository. And this data model is what they use to share this data. Interoperable. This I in FAIR is that that the metadata uses standard sort of forms of knowledge representation. Again, these are metadata models, and that they use vocabularies and terminologies that are fair. And so when you're using terminology, you have to be careful of how you describe things in your data sets, right? And that you have the appropriate data dictionary. So for example, you may have three different labs studying, a rat model of type 1 diabetes. You have, different notebooks, where you have, age, is written in different ways. So one is an age range, one is just adult. So what does adult mean in this context? You have, an organism classification. One where you have a number for animal, one where you have a subject with some cryptic text, and then the organism some more cryptic text. And so this is how do you make sure that when you're talking about these organisms that they are the same organism or not. With genes, there are again different standards for gene nomenclature. And so which are you using internal shorthand, or are you using NCBI's gene names? What are you using here? And then also for anatomical locations, people use shorthand. They use specific information. So for example, in data set 3, I don't even know if this is related to kidney. It could be cortex in the brain, right? So again, that's a lot of times when data is collected within an experiment, there is a scope for that experiment. So writing down information in the scope of that experiment, when someone else is looking at it from outside, is not unique, right? It becomes ambiguous. So there are a number of best practices, especially when, you're talking about various aspects. So for reagents and tools, there are these research resource identifiers, which I'll mention in just a second. Genes, there are again standard gene nomenclatures to use. And then if you're talking about anatomical location, make sure that that is well specified. For example, that it isn't cryptic shorthand or something that is ambiguous to someone coming from the outside. And you can look at you can get a lot of the these terms from ontologies such as Uberon or the foundational model of anatomy. So again, if we were to actually associate these with, for example, the RID, the gene ID, and the actual anatomical identifier, then it's very easy to know that data sets 1 and 2 are talking about the same thing, whereas data set 3 is not, right? So this is actually the cortex in the brain. So for RIDs, you can actually go to dkNET to our resource reports, and here you can find information on tools, cell lines, antibodies, organisms, and more in terms of the RID and information about that resource. And so again, recording these early on. So for example, when you if you're using specific antibodies, when you actually purchase the antibody, a lot of antibody vendors have the RID on their site. You can actually record that right then and there, so that you have that later on, instead of having to go back and then not being quite sure what RID that you should attribute. So again, this is all in all part of this these data management practices. And then reusable. This is the last bit of fare. And here we get to the richness of the metadata, right? So that the data metadata richly described with a plurality of accurate and relevant attributes, and these metadata data meet domain relevant community standards. And really here, this is about thinking about where your data will end up, when you start your experiment. And that's why NIH has placed significance of the data management portion of the data management and sharing plan, right? So that when you're already beginning to think about an experiment, that you're thinking about which repository this will end up in, because certain repositories will require certain metadata. And it's good to know that before you hit the submission process, because if you haven't been collecting that metadata, that becomes then, a significant bit of effort to sort of go back and backfill all that metadata. Whereas if you know upfront what a repository requires, you can be collecting that metadata, from the start. And that will make it much more streamlined, when you're doing the submission process. So in terms of repositories, through dkNET as well, we do have a list of repositories, domain-specific and general purpose, that related to NIDDK, and also recommended by NLM at NIH. And we're also working on some additional tools, to assist researchers in this process. And again, data repositories, they're all different. There are hundreds of them across biomedicine. There are different types. So there are, domain data repositories that are for specific data types. So for example, metabolomics data. There are domain-specific repositories. So repositories, focused on spinal cord injury. There are generalist repositories, right, which aren't affiliated with any specific domain. And then, of course, many institutions have their own repository that can be that researchers can use. This is again something that is a landscape that researchers are going to have to be able to navigate. And as I mentioned in the prior slide, through dkNET, we're trying to make some of those things more easy to navigate. The other aspect is what standards are you using or should you use? So again, if you are looking at certain repositories, they list their standards, and there's also a really good resource out there, fairsharing.org, which provides information on repositories, the standards they use. And so this is something that you can definitely use as a resource when thinking about standards for the data that you're collecting. And then for the data management and sharing plans, there are a number of resources that are being made available. So again, we list these through dkNET, but there is the DMP tool from the California Digital Library. There's MIT Libraries has a number of training aspects to that. And there's also the Mantra resource. So again, there are a number of these that are available, and you can come to our site to find those. And the other thing is that data is becoming more and more viewed as a research product itself, right? Which means that it should be a citable product of research, and this is tied into data citations and data citation principles that journals have actually picked up. So most data in repositories is identified via DOI, just like journal articles. So there are a number of groups that mint DOIs or allow people to mint DOIs for data sets. The data citation, a full citation, looks very similar to a journal citation. Additional things you specify potentially the internal identifier for the data set at the repository, plus also the DOI. But once that is once that DOI has been published and is used for cite, then just like your citations are tracked for your published literature, these citations for data sets can be tracked the same way, so that you can see who has been using data sets that you generated. And so these data citation principles are critical for the use of the data, but also to tie it into sort of the scholarly commons. So I bet a lot of people are feeling overwhelmed, but again, it is a partnership, right? So again, you're not alone in this, right? So researchers responsible for good data management, make sure you're collecting good metadata, you're aligning the standards, and that you've looked at and already figured out sort of where you're going to be pushing your data. A lot of the other aspects are in terms of the persistent identification, making sure the metadata is available, that there are clear licenses, the data citation, that's done by the repositories and registries. So again, you're not you should try to look at this as a partnership. And then there are other aspects to that, like dkNET. where we in the DK community try to help researchers, navigate, these areas. So again, having trouble, ask dkNET. We do have full sections on, the data repositories, best practices in managing research data. So again, NIH and projects like ours are making more and more information available. And so again, this is all part of a cultural change in biomedicine, where this sharing of data is moving along a line. The genomics community has been sharing data in this way for a much longer period of time. There are other communities that are well along their way. There are others that are not. So this is all we're all sort of in this boat together. So wanted to thank everyone, and I can take some questions, and then at the end of the slides there's a nice little YouTube video which data sharing and management snafu in 3 short acts. So I don't know if anyone's seen this, but this is a link that is in the is in the slides, and this is actually, I think, gives a pretty good overview of why metadata, persistent identifiers, and sharing are important. So just wanted to mention that, but I will now take any questions. I will stop my share.
Hi, I have a quick question. Can you hear me?
Yes, yes.
So for the purposes of the proposal, can we assume that if we find any data through the dkNET and the links and the tools that these would meet the FAIR standards?
For most, if data is coming from a data repository, it usually does. Again, FAIR is a continuum. So there is the issue of the sort of richly described metadata, right? So you can have the high level metadata that's, so you could have a data set that comes out of Figshare or Dryad, one of the generalist repositories, and it has high level data descriptions, but may not have, some of the deeper metadata annotations. So again, FAIR is an aspiration. So we're all trying to move to FAIR, and so the different repositories are different levels of FAIR. So that's really, for example, if you get a spreadsheet, tabular format data out of a repository such as Dataverse, it's going to have a lot more metadata around the data dictionary, right? What do the columns represent? What's in the columns? Just like if you had your data in REDCap. For example, if you're storing your clinical data in REDCap, it has a data dictionary. And then, when you export that data, you can also export that data dictionary. But sometimes, if you're using a generalist repository, they may not require that data dictionary. So then it's on the researcher to know that they have to provide that information. So a lot of the F and the A definitely are, if you put it in a repository, F and A are sort of fully supported there. The I and the R is, again, this continuum, so how much metadata description the repository requires.
All right, thank you.
Okay, thank you.
Thank you.